Good day, everyone. My name is Dexter Daiz Mahinay with my partner, Lopez Jr. We are from Animal Science 1B. In today's video, we are going to discuss to you our assigned topic, which is soil analysis and soil testing. Before we proceed to our report, we first define what is soil analysis and soil testing. Soil analysis is the set of various chemical processes that determine the amount of available nutrients in soil, but also the chemical, physical, and biological soil properties important for plant nutrition of soil health, while soil testing provides an accurate assessment of the soil's fertility to make fertilizer recommendations. With an increasing awareness of fertilizer effects on environment quality, soil tests also can be used to determine where fertilizers or manure should not be applied. In that simple explanation, the definition of our topic, we are sure that we gain a little knowledge on why do we need to know the purpose of soil analysis and soil testing in our studies. Objectives: explain the purpose of soil analysis and soil testing. Describe soil sampling techniques for laboratory analysis. Outline the procedures involved in soil correlation and calibration. Explain how interpret soil test data followed by fertilizer recommendation. In these objectives, we come up and we find answer after discussing the soil analysis and soil testing, sampling techniques, correlation, and soil test data followed by fertilizer recommendation. Introduction: fertilizer nutrients use efficiency by field crops depends on very reliable and economic fertilizer recommendations. The knowledge of the quantity of soil reserves of plants' nutrients ensures that adequate amounts of the nutrients are added to the soil for maximum crop yields. Recommendations are based on experimental work which includes soil analysis, plant analysis, and soil testing programs. Blanket fertilizers recommendations must be avoided. So, fertilizers are very important to plants and farmers because plants needs nutrients from soil and that nutrients will help the plants to make their own foods with the help of water, sunlight, and carbon dioxide through the process called photosynthesis. Farmers uses fertilizers to their crops, especially in their crops places that has low of nutrients of soil. They uses it to supply the needs of nutrients by the plants to have better harvest at the end. Farmers, engineers of the soils, researchers of soils are conduct studies about the soils in their respective area, which they test the soil and plants if the soil has enough nutrients for plants needs, and if plants has too much nutrients, the farmers not uses fertilizers because too much fertilizers of plants can kill plants. Purpose of soil analysis. Number 1. Generally done to predict the behavior of the soil when put into various uses like cropping, road construction, and so on. That is, evaluation of soil productivity is achieved. Number 2. The analysis may be to discover its origin, its composition, constituents, or effect of changing environment. Number 3. Routine soil analysis is useful for advisory purposes. Prediction of probability of getting profitable response to fertilizer application. Number 4. Analysis may be for the purpose of research, whereby the analysis is specific. Number 5. In soil survey, the analysis may be to classify soil, grouping of soils into classes for the purpose of fertilizer and lime recommendation. Data provide guides for plant nutrient recommendations. Number 6. Chemical soil analysis investigates the capacity of the soil as a medium for plant growth. However, other factors such as climate, microbiological conditions, structures, and other physical properties affects plant growth. 7. The determination of a specific soil condition that may be improved by the addition of amendments or cultural practices. Number 8. to develop appropriate methods for estimating the availability of a particular nutrient, detecting plant nutrient uptake and responses to various cultural treatment. So, the purpose of this topic is to know the behavioral of the soil when it put to various uses like cropping, reconstruction, settlements, building constructions, and many others. In this analysis, we discover the origins of soil and its composition by affecting its changing environment. After studying of soil analysis, we gain a knowledge about soil advisory, in which we know on what application of fertilizer we use in a prospective lands for plants needs. After these studies, we also know on investigating the capacity of soil if some factors that affect soil like climate, microbiological, microbiology conditions, structures, and other physical properties that affects plants growth and find solution in this problem to supply enough perfect fertilizer to soil for plants needs. To know how to improve the quality of soil using amendments or cultural practices to improve soil texture or fertility. And lastly, to know the appropriate methods for estimating the availability of soil in particular nutrients in plants and to detect the plant's nutrient in their responses to various cultural treatments. Characteristics of soil analytical methods for practical advisory work. Number 1. The methods must be precise, reliable, simple, and cheap. Accuracy refers to the correctness of the method, while precision refers to the producibility of an analytical result. So, they must obtain a correctness of ways in analyzing the soil. Precision, reliable, simple, and cheap methods that will help them to be accurate and productivity result in soil analysis. 2. Methods for determination of available nutrients take into recognition that the available fraction of the total amount of nutrients in soil is not uniformly available. So, they must have the correct knowledge of knowing the different fertilizers in order to use in the soil and what kind of fertilizers needed and its amount. 3. Methods for determination of available nutrients is designed to estimate either the total available fraction or only part of it. The different forms are interrelated. Plant takes up nutrients from the soil's soluble water, soluble form or intensity factor. The maintenance of the high intensity factor depends via its changeable form on a high content of easily mobilizable reserves capacity factor and on the rate of transfer from one fraction to the other. It means that they must know the different ways in determining the nutrients in soil, and all of these are connected to each other. They must obtain the different ways in determining the nutrients needed by the plants. 4. Choice of methods depends on the special behavior of the nutrients in soil and the production level. A. Cationic nutrients have high exchangeable fraction, which should therefore give a fair estimate of the available fraction, especially if the reserves are relatively low. B. At the low production and fertilization level, plants are forced to use largely mobilizable soil nutrients, and the methods must place emphasis on the reserves. High-yielding plants at high production level should have a good supply of easily available nutrients in soil, since these crops are selected for high production and not for high utilization of nutrients reserves. So it means that they must have the skills in choosing a methods that depends on the behavioral nutrients of soil in using different ways to have a good production. If the nutrients of the soil cationic has the high, it must share a nutrients to side in which nutrients is low, especially if the reserves nutrients per plants is empty. To have a perfect nutrients to the soil that the plants need, but if the plants has low production, the farmers must use different ways like using the organic and inorganic fertilizer in the soil to supply the needs of plants and to have a productivity of crops. When the plants have high production, it means that it has a good supply of nutrients from the soil. Because the result of plants has the high production, it means that a high a farmer should use fertilizers. 5. For several micronutrients Fa, Mn, Zn, Cu, the chelating reagent DTPA, diethylene, trimine, pentacetic acid seems to be quite useful and it applied in several countries. So it means that for some several micronutrients like iron, manganese, zinc, copper, and chelating reagent DTPA, diethylene, trimine, pentacetic acid are very known and useful into soil nutrients in different countries because it is exporting to different countries and many farmers using these fertilizers. 7. 6. Chemical soil analysis can be done at any time of year unlike field tests which must be done during the growing season. So it means that they do chemical soil analysis anytime in the year so that the nutrients of soil can be used by plants, not just like field tests who make the process in the time that the farmers do a planting. 7. Evaluation calibration of soil methods. Calibration is necessary in order to select the best analytical methods for a given soil and production condition. So it means that the calibration methods of soil is establishing a relationship between a given soil test value and the yield response from adding nutrients to soil as fertilizer. Relative yield is obtained from fertilizer experiments where the nutrient tested is the main factor and solely responsible for the yield obtained. So relative yield, it means that is where we can find the result of fertilizer experiment by the plant's fruits after harvesting it if it is good or bad production. The fertilizers experiments are responsible for the plant's growth. Nutrients uptake of content. Plants can be used if fertilizer experiments are difficult to carry out. There is not uniform field of deficiency, especially for trace elements. In the range of latent deficiency, the optimum supply and content of nutrients in plants reflect the contents available nutrients in soil and can therefore be used as an index of calibration. So it means that nutrients uptake of contents in which votes as inorganic ions from soil solution. The rate of nutrients uptake is independent of the rate of water uptake, but the concentration of nutrients at root surface depends strongly on soil water content. Final evaluation. The comparison of soil instruction as R2 value present, the R value often used in much less adequate and less intelligible soil methods should have times 2 valued at the least 50 to 70% in order to be useful for manganese or Mn. So final evaluation, the soil testing takes place, which the soil after testing is comparing to soil to other soil like in figure below. Table 16.1. Calibration of soil tests for available manganese and Egypt. Correlation with Mn or manganese content of soybeans. Metwali et al. 1973. So we have methods. R2 equals the value of percent. So 0.1A1 phosphoric acid, we have the value of 28. 0.05 M or manganese EDTA, we have the value of 34. 0.5% hydroquinone in MH4 acetate, we have the value of 69. 0.025% hydroquinone in NH4 acetate, we have the value of 63. The method with the highest R value is the best method, especially if the regression is rather step. Correlation could be improved by using several soil factors in addition to the available fraction, multifractional correlation. The calibration of soil method should not be done with one country but also on the regional basis, such as in the uniform area as the arid zone of Middle East. A good cooperation can save a lot of individual calibration work. So correlation is the relationship between the amount of nutrient extracted from the soil by the laboratory test and nutrients uptake by plants in the greenhouse or field or crop yield. Sometimes, the relationship can be established only for one nutrient in one crop and on a particular group of soil. Correlation are therefore of the ways and testing ability of soil in which happen not only in one kind of soil but in different kind of soil. This study happens not only in one country but in different country where it has a different kind of soil. Interpretation of soil test data. A. The functional curve fertilizer response curve between soil data, available nutrients, and yield data in order to deduce the optimum content of available nutrients required in the soil for maximum yield. So it means that functional curve, fertilizer response curve, available nutrients of soil, and yield data are the ways to determine what nutrients obtained by the soil and to supply the nutrients needed by the soil to use by plants to have a better result of harvest at the end. B. The amount of fertilizer required to cover the difference between a certain relative yield and the maximum yield, or in practical terms, the amount of fertilizer required in order to reach the optimum soil nutrient content. So it means that the fertilizers are very important to soil because the plants depends on the soil nutrients and it uses the plants to compare the result of harvest in the end using the same crops and to reach better harvest. C. Interpretation can be made in form of mathematical curve or in a less sophisticated way in terms of fertility classes. The criteria for soil fertility classes in Nigeria are as follows. I. Low, the value below critical level, which implies application of high dose. Double I. Medium, the range above critical level available response to fertilization is expected to apply one-third to one-half dose. Triple I. High, the range where response in unlikely and fertilization may not be necessary.
Nitrogen is defined in terms of total nitrogen. Phosphorus in terms of Bray1 or Bray2. Potassium in terms of NH4, OAC, exchangeable potassium and organic matter in terms of Fockley and Bach total organic matter. Table 16.2 Rating for soil fertility classes in Nigeria. Nitrogen total. We have here the low, medium, high, and the phosphorus Bray1 and Bray2 low, medium, high, and the potassium with low, medium, high. Data is example of organic matter, soil pH, the magnesium, soil sampling procedure, area per sample, composite sample, and subsample. Depth of sampling. Usually, we sample to the depth of tillage because most of nutrients are concentrated in the surface, where most of organic matter are found. The depth is usually about 15 cm. There should be at least 2 composite samples per one area. Tools. Use sampling tools such as soil probe. Second, soil auger could work itself around small stone of gravel. Spade or shovel. We excavate with spade to a growth depth of 15 cm and use cutlass to cut off 2 cm of soil. The soil sample is put into plastic bucket or bags. After taking soil from field, we dry and mix all individual samples together very well. We take about 500 grams or 250 grams for chemical soil analysis. Fifth, map of the area. We indicate the number of samples per area. We should draw a map to locate the area of this sample. Next, container must be free contamination and keep clean. Next, area to avoid in sampling. Avoid any area that is obviously different from the rest of the area, unless the odd area is large enough to treat as a separate sampling unit. Such areas are: first, eroded area, where the top of the soil could have been eroded away. First, eroded area, where the top of the soil could have been eroded away. Second, depressing or wet area. There will be a different exudation reduction in the soil, thus affecting soil analysis result. Area adjacent to animal burrows. Area where Aeron residents have accumulated, such as empty cocoa ponds which swell up and decay in the soil. Areas under large tree, especially oil palm. This applies to clean areas where we have isolated trees, where there will be accumulation of OM to swell up CEC and nutrient of the soil in that area. 6. Dead furrows. In the field in general, we must avoid bulking together sample cores from usual places. Once we have the representative sample, we take it to the laboratory for analysis. Soil test correlation. The purpose of soil test correlation is to develop an extraction procedure for a particular nutrient. Therefore, we estimated our result and tried to correlate the extractable form of nutrient with the amount of extracted chemically. This procedure is also known as soil test correlation. The area to be sampled depends on the area for which the test is done, but there must be a minimum of 20 to 30 sample for the area. Suppose we are extracted for P. We grow the crops in the greenhouse supplying other nutrient except P. We may grow maize or fast-growing grass, which is grown for about 6 to 8 weeks. The crop is harvested, then dry weight of plant material and the concentration of P in the tissue are analyzed from dry weight to concentrated P. The uptake of the nutrient is calculated. Uptake of P equals yield x concentration. P is in tissue. The uptake equals available P in the soil. The next step are to find reagent in the laboratory that would extract from the soil the same amount of P that was available to the plant. This is an ideal condition, but we must be able to find the extractant that would extract an approximately amount as an plant uptake of the available form of the nutrient. First, NH4 CAC. Second, INHCl. Third, 0.INHCl. Bray test 0.03 N NH4F in 0.5 N HCl. This measure P bounded to A1 and Fe compound by forcing with Al and Fe particles, thus releasing H204. After using so many reagents, we draw a graph of available P against the amount extracted chemically, making separate a graph for each reagent used. Each soil should be properly characterized, physically, chemically, and taxonomically, in order to facilitate the extrapolation of soil test information to similar soil in similar environment. Each essential economic crops of the area and essential nutrient elements should be calibrated because crops have different nutrient requirements. In Nigeria, mixed or multiple cropping should be considered in soil calibrating works in additional monocropping step in conducting soil test calibration. A. Find a soil that has low level nutrients so that is used as starting point of nutrient additions. B. Select at least 5 points of curves so as to define shape of the curve. A soil may already have 5 kg per hectare. From soil analysis, we would add in this order giving this curve. P added and yield obtained. For maximum yield, maybe should need to add between 20 to 40 kg for that soil that fix more P. Therefore, we carry out this type of experiment on different soil on at least 4 to 5 locations. After maximum yield level, it takes a larger amount of uptake to have further increase yield. We may need to find out the toxic level. The aim of soil calibration data is to know how such nutrients should be added to have maximum yield. We have to do a replicated experiment for 3 to 5 years in order to take account of variations of weather and OTH. D. We have to find out how much nutrient should be added to rise level of available nutrient to a certain amount because part of the added nutrient will become fixed. Fertilizer recommendation. Soil test information should be extended to the farmer who should be instructed on time, rate, and method of fertilizer application. The information should be also include improved crops, varieties available and other production management practices. Means of disseminating the package of recommendation include radio, television, newspaper, field days, poster, agricultural shows, meeting, film shows, bulletin, and leaflets, training courses, demonstration, and personal contact. To make fertilizer recommendation much more reliable, especially under multiple cropping system, the use of fertilizer factor had been developed. Fertilizer factor is the amount of nutrient element required to raise the soil test level by 1 mg to 1 kg soil. The means and range of fertilizer factor had been developed for some nutrient in the savanna region of Nigeria. They are P equals to 3.1, close up in parentheses 1, 7, K equals to 4.5, Mg equals to 3, Zn equals to 6.4, F equals to 2, Cu equals to 2, and Mn equals to 0.8. The following is used as a multiplier of calculating fertilizer requirement after determining the critical soil level. For example, if Bray IP soil test of a field is 10 mg to 1 kg and the critical P level is 15 mg kg, fertilizer recommendation for P will be X 3.1 equals 15.50 milligram P per kilogram soil. Multiply by a factor of 2, convert to kilogram P per hectare, 15 milligram per kilogram times 2 equals 31.0 kilogram P per hectare. Employing only the critical level concept, Kate and Nelson model would have given of x2 equals 10 kilogram P per hectare as the fertilizer recommendation. This quantity of phosphorus will not be sufficient to saturate the exchange complex of the soil and releasing P for the crop. Conclusion: the aim of fertilizer use is to supplement the natural soil supply for optimum crop yield. It is therefore imperative for the soil scientists to have true knowledge of soil analysis and soil testing data. Interpretation: it should be emphasized the soil alone is not equivalent to soil testing. Proper interpretation of soil data leads to valid fertilizer recommendation for optimum yield and for satisfactory content of trace elements and minerals for livestock feeding. The major purpose of soil analysis, the research component of soil testing, data interpretation, guide, and recommendation have been carefully described.
Summary: in this unit, we have learned that 1. Soil analysis is just only a part of soil testing which is carried out for various purposes. 2. Several analytical procedures and instructions are available for a particular nutrient element. 3. Soil sample collection on the field is done carefully to adequately represent the field. 4. Soil testing involves soil test correlation in the greenhouse and soil test calibration in the field. 5. Both correlation and calibration data are employed in making fertilizers recommendations to farmers. Reference: Further readings. Adoye, G.O. 1998, Interpretations of Soils and Plant Analysis for Agronomic Use. Soil and Plant Analytical Laboratories Network of Africa, SPAMNA. Brandy, M.C. and Ray R. 1999, The Nature and Properties of Soils, 20th ed. New Jersey, Prentice Hall. Westerman, R.L. 1990, Soil Testing and Plant Analysis, 3rd ed. Madison, Wisconsin, Soil Sci. Soc. Amir.